Hello, thank you for joining. My name is Kit Fuhrman. I am the product manager for the GeoMx RNA assays. Today, I would like to introduce our Cancer Transcriptome Atlas, which is an essential tool for spatial genomics and oncology research. So genomics research has evolved quite considerably over the last 15 years. Bulk gene expression has taught us much about tumor biology. Just like that fruit smoothie on the left, we've had to homogenize the tissue for this to work. Even so, these gene expression signatures has taught us a lot about the state of the tumor, about how it's inflamed, and how we can inform different decisions. So the next iteration was single cell biology, where we were able to pick apart and figure out all the different pieces of fruit that went into the fruit smoothie. We can discern different cell types and different phenotypes and functionality in these cells, but putting it back together is where we struggle right now. Putting it back in the original framework and the original architecture of the tissue is a very difficult task. This is where the power of the digital spatial profiler comes into play. It allows the user to see the spatial landscape and quantify expression across important regions of interest. Today, we are introducing the Cancer Transcriptome Atlas, or what we call the CTA, combined with the NGS readout for the GeoMx DSP platform. The CTA is the first GeoMx assay to leverage the almost unlimited plex of our new NGS readout chemistry. We've built reagent probes sets that allow an almost unlimited number of targets to be quantified simultaneously. With the CTA, you can measure 1,834 different mRNA gene transcripts throughout the tumor, immune system, and the microenvironment. Importantly, our technology is not limited by high expressors. DSP enriches for transcripts that are critical to your research, regardless of expression levels. And you can map these gene expression values directly to regions within your tissue. So when designing this probe set, we began by adding in all the important immune targets. The immune system plays a critical role in oncology and in many other diseases. So we included genes involved in the innate and adaptive immune systems. Any researcher studying the immune system, whether in infectious disease or oncology, will find these targets informative. We also layered in targets from our key encounter panels, including the pan-cancer pathways and IO360 panels. We added clinically relevant gene sets, and we added in metabolic genes. There are a considerable number of metabolic gene targets in this panel, and it allows the researcher to study the metabolic states of the tumor and the immune system. Importantly, we also added in a lot of information and gene targets through community input. We mined both the literature and other presentations and online gene sets in databases to make sure that all the important informative targets were in this panel. Finally, we fully annotated the probe set. This allows the user to bring context to every gene in this atlas. The CTA includes genes to identify critical immune cell types, such as T cells and macrophage subsets. We've also added gene sets that offer insight into immune biology, such as the tumor inflammation set and the PAM50 gene signatures. The Cancer Transcriptome Atlas encompasses over 95 different biological pathways and has many annotations built in. Having all this great content in one place is made possible by our unique chemistry. Our probes hybridize directly to transcripts within the formalin-fixed paraffin-embedded tissue section or your fresh frozen tissue section. The readout portion, which we call the DSP barcode, contains a readout tag sequence for every target and a unique molecular identifier for every molecule. We also included the TrueSeq primer regions to make the readout compatible with an Illumina sequencer. The photocleavable linker between the probe and the readout portion is a key part of the DSP platform. We use UV light to collect the DSP barcode for downstream analysis for specific regions of interest. Any project begins with staining a tissue section with the Cancer Transcriptome Atlas probes and a set of up to 4 morphological markers, including a DNA state. This is step 1. The morphology markers can either be antibodies with fluorescent labels or RNA scope probes to visualize individual transcripts. The markers allow us to visualize the tissue sections and guides the region of interest or ROI selections. This is step 2. So I would like to take a moment just to talk about the RNA scope probes that we've integrated into the GeoMx workflow. Visualizing and understanding the landscape of every tissue is critical to connecting the genomics information to spatial context. RNA scope is an integrated tool to visualize single transcripts. You can visualize up to 3 different transcripts, and you can choose from our list of predefined RNA scope virtual kits, or you can build your own combination. RNA scope is a unique tool for selecting regions on oftentimes difficult to target gene targets such as TGF-beta and FOXP3. So I'd like to give you an example of how we go about selecting regions of interest. Here we have non-small cell lung carcinoma tissue section, and it's been stained with 4 morphological markers: DNA, CD3, PDCD1, which is the transcript for PD1, and CD274, which is the transcript for PDL1. So these are immune checkpoint genes. And so we've scanned the whole tissue section, and then we've zoomed in and selected individual regions of interest. We've used our morphology stain to select 3 different region types across the whole tissue, so multiple regions of the same type. On the top, you can see that we've selected regions that are high for PDCD1 in the stroma, and then regions below which are in the tumor, which are low for PD1. And then on the right, we've selected tumor regions that are high for PD1. So in this way, we can categorize the different regions that we're selecting. DSP, importantly, allows us to study the transcriptional profile of each of these regions independently. So now that we've stained our tissue and selected the regions of interest, the machine will then illuminate every single region. The barcodes, the DSP barcodes, will come off of the tissue. They will be collected by the instrument and deposited in a well, in a 96 well collection plate. This process is then repeated for steps 3 and 4 for every single region that the user has selected. At this point, the collection plate is then processed in our library prep and sequenced on the Illumina sequencer. Now that each region is in one well in our collection plate, we can take it and process the DSP barcodes. NanoString provides all the reagents you need to perform the library prep. Then once it's sequenced, we provide all the tools needed to take the data from FASTQ files to analyzing the data. To construct the library prep, NanoString has developed the GeoMx seq code reagent to process and collect the DSP barcode oligos from the CTA assay. We've built an 8-plate set of universal dual index adapters. These indices are properly color balanced indices so that you can process DSP barcodes in the order they are collected. No need to cherry pick wells. Then the system will actually identify how to set up the library prep. We use the dual indices to map the DSP barcodes from the illuminated regions to the resulting genomic CTA gene expression data. We also provide the PCR enzymes with the seq code. Along with the well-specific primers, the PCR adds the P5 and P7 regions to the library constructs. Once the library is generated, you can pull all of the wells together and run them on an Illumina sequencer. You can multiplex a minimum of 4 wells or all 768 wells from all 8 unique plates. The seq code library prep are compatible across a wide range of Illumina sequencers. The number of reads needed depends on the size of the region selected. For a typical region, that is between 2 to 3 million reads. For each slide with a typical number of regions, between 12 or 24 regions, You'd want to plan for 30 to 50 million reads. Only single end reads are needed, but you could certainly run the library with paired end reads if you'd like. Once the library has been sequenced, NanoString provides the tools to process the FASTQ files and transform them into digital counts. You take the FASTQ files from your Illumina sequencer data run, and then we have an automated data processing pipeline that then converts them into these DCC files. You then will upload these DCC files into the GeoMx platform, and it will connect the gene expression data back to each specific region of interest. You can now build your analysis in DSP in the DSP Data Analysis Suite. On the left-hand side, we have the Slide Explorer. We can add in all the slides from our study. This is a critical part of the platform where you can build these data analyses from multiple sections and multiple slides. So you can build an analysis across large cohorts of samples, from clinical trials, data sets to large internal studies. The software has all the tools to check the quality of your data, normalize your reads, and analyze the data for interpretation. The middle section keeps track of any changes you make to the data and has a list of all the gene annotations that we talked about. You can also visualize your data in interesting and informative ways. Importantly, the software is interactive and the genomics data is always tracked back to the region of interest and the segment that you're working with. So going back to our non-small cell lung carcinoma samples, here we have these two region of interest types, PD-L1 high and low regions in the tumor. The DSP data analysis suite can analyze all of the similar regions and then compare the expression profiles between the two region types. So here we're looking at, on the right is a volcano plot comparing the CD274 high and the two CD274 low regions. And you can get a list of all the differentially expressed genes between those two region types. This is just one sample, but you can expand this analysis across a large data set of samples. I would also encourage you to check out our colorectal carcinoma white paper that uses the Cancer Transcriptome Atlas to study the differences between two different main types of CRC sample types, one which is MSS and one which is MSI. These two different clinical annotations have very different outcomes, and we use 3 different profiling types to analyze this set of samples. We choose different region of interest selection methods across multiple samples to analyze the immune system and how it's changing both across the individual samples and between these two different, very different sample types. So in review, the Cancer Transcriptome Atlas has an expansive collection of content, over 1,800 genes across 95 annotated pathways. It is very comprehensive and includes targets in tumor biology, immune response, and the microenvironment. It has critical gene signatures, and you can actually add in targets of interest. It has a minimal sequencing requirement between 30 and 15 million reads per sample. You can try it now in our Technology Access Program, and it's going to ship in August 2020. Thank you for joining this presentation.